I'm really happy and would like to introduce you to our host of this webinar, Natia Gut-Biazini, President of the Friends of German-Speaking Switzerland and Governor. Nadia, the floor is yours.
Thank you, Mara, for organizing so many webinars. Good evening, everybody. Long-term friends and fresh ones, fresh friends of the Hebrew University. I'm very glad to open this evening and webinar by Professor Yossi Buganim, who is going to inform us about regenerative medicine. Yossi Buganim, an associate professor at the medical school at Hadassah. Hadassah is at the Hebrew University, and a favorite of our Swiss-German friends, is specialized in reprogramming cells, as I learned. Reprogramming the nuclei of the cells, and furthermore developed new techniques in order to generate stem cells from skin cells. That's what I learned through the words. A few years ago, I had the opportunity of getting to meet Yossi on the occasion of his presentation upon receiving the Zellman Cohen Fellowship, which is given between Sydney and the Hebrew University. And the term for this fellowship is to improve the human condition. In 2019, together with a friend and member of our board, Noemi, we visited Yossi's lab at Hadassah. We got to know a very enthusiastic crew of students and PhDs and postdocs, as far as I know. Of course, they were all hardworking, but they also told us that Yossi is always helping them when they do not know how to move further or solve a problem. So we really like them. I'm equally thrilled that Dr. Therese Lüthi will lead us through the evening. Therese is an excellent science editor and journalist of the Neue Zürcher Zeitung am Sonntag, on Sunday. This is a newspaper which is edited in Zurich and is the best newspaper in Switzerland. It has a very good science edition. Therese has actually published in the field of scientific journalism for about 30 years. That's what we found out today together.
Almost.
True? Almost 30 years. Therese got her PhD in neurobiology at the Biozentrum in Basel, which included also a period at the Rockefeller University in New York, and perfected her studies in science reporting with an MA at New York University. Together with her family, Therese lives in Zurich. She grew up in the States and in Frankfurt. Please, Therese.
Thank you, Nadia. Good evening. So I'm very happy to introduce our speaker today, Yossi Buganim. He is associate professor at the Hebrew University in Jerusalem. Yossi received his undergraduate degree from Bar Ilan University and then got his PhD from the Weizmann Institute of Science. For his postdoc, Yossi went to the Whitehead Institute for Biomedical Research at MIT, where he worked on new nuclear reprogramming techniques, basically trying to understand what it takes to convert skin cells into other medically relevant cell types, such as heart cells or neurons. He is currently working to improve these techniques and to develop new paradigms of cell conversion for future medical applications. Yossi Buganim is an extremely promising young scientist. He is the recipient of various awards, very prestigious awards. He was nominated as an International Howard Hughes Medical Institute's scholar. That makes him one of only 6 young scientists in Israel and of 41 young scientists all over the world ever to have won this award. Yossi also received the Stem Cell and Regenerative Medicine Award by the prestigious magazine Science. Yossi will now speak for about 30 or 40 minutes, and he will then be happy to take any questions. So if any questions pop up during the talk, please write them into the chat on the right. So Yossi, we're all excited to hear your talk now. The screen is yours.
Thank you, Therese. So just let me put my presentation on. Okay, so good evening, everyone. I hope you see the screen, my presentation. So first, thank you, Therese, for the nice introduction. And I'm really excited to be here and to present my research in front of this wonderful audience. And before I start my talk, I really would like to thank also Nadia Goods for the invitation and Mara for organizing this event. Thank you very much for that. So when I start giving a talk, I always like to start with the motivation. Why are we doing this, this specific research? So we have two major motivation in the lab. The first one is a lack of life-saving organ donation. And let me show you some statistic about that. So currently, and this is true only for the US, there are about 120,000 people who are waiting for life-saving organ transplantation. Every 10 minutes, a person is added to the list, and on average, 21 people die every day while waiting for organ transplantation. And organ transplantation really works. You can see here in this graph that the survival rate of 5 years after transplantation is really high, about 80%. But the problem resides in this graph. Here you can see the increase in the demand of organ transplantation every year, and here is what we can give them. So obviously, this terrifying statistic urges the field to find a solution and to grow cells outside of the body. And I would like to start my talk in the end. And just before that, I forgot to mention the second motivation is actually aging-related diseases. As you all know, as we age, people encounter many aging-related diseases like neurodegenerative diseases, Alzheimer's, Parkinson, diabetes, and cancer. And if there is a way to prevent this, this will be really amazing. And as I said, I would like to start at the end of the talk, which is our vision. How I see the future medicine in 50 or even less years from now. So in my vision, I see a patient with a specific disease, for example, Parkinson or Alzheimer, come to the clinic, and then the physician take a skin biopsy from this patient, and then the lab converts these skin cells into different clinically relevant cell types. So if we are talking about Parkinson or Alzheimer, these skin cells will be converted into neurons, and then we can transplant back this neuron into the same patient. We also know how to fix a mutation in these cells. So if we know the problem of the neurons, we can fix them. We can fix the mutation before we transplant these cells back to the same patient. So this is one aspect of our vision, which called regenerative medicine, the ability to grow cells outside of the body. The second vision is related to aging and about rejuvenation. In my vision, I see old people come to the clinic, then the physician just take a blood test from them, isolate all the stem cells, all the very primitive cells of the blood, and then manipulate these cells to become younger, and then transplant these blood stem cells into the same patient. What we are actually doing here is rejuvenating our immune system. And as immune system is the cause and the solution for almost every aging-related diseases, I believe that by rejuvenating our immune system, we can really prevent devastating diseases such as Parkinson's, cancer, and Alzheimer. So this is the end, but let's start in the beginning. So what is regenerative medicine? Regenerative medicine is the ability to take cells and grow them outside of the body. We cannot just go to a specific patient and take his cells, right? We need to find a source of cells that will grow forever and will be able to give us any cell type that we want. And we have such a source. The major source for the entire field of regenerative medicines are embryonic stem cells. So what are embryonic stem cells? Here we can see embryonic development. Here we can see a fertilized egg that starts divide and it becomes 2-cell stage, and then 4 cells and 8 cells, until we can see a clear early embryo that's called blastocyst. Now I'm moving here to this plot. This blastocyst, we can illustrate it here with 3 colors. This blastocyst is composed of 3 compartments. The first compartment are the red cells, and these cells will give rise to the fetus. And if we will put them in the dish, they will give rise to embryonic stem cells. And these are the cells that we are looking for. We have the green cells, which will give rise in the embryo to the placenta. When we will put them in the dish, they will give rise to placental stem cells. And we have the blue compartment, which called primitive endoderm, and these cells will give rise to the yolk sac stem cells. So these are the 3 major cell types that build the early embryo. And here we are talking about 64 to 128 cells, and this happens 7 days after fertilization. So if we take this embryonic stem cell that we isolated from the embryo and now grow them in the dish, we can put different cues, different signals, and force them to become any cell type that we like. For example, we can give them several cues that will force them to become neurons, or intestinal cells, or pancreatic cells, or liver cells. So in theory, this should solve all the problems that we have in the field. We have here cells that can grow forever. Once I have enough amount of cells, I can give them a specific cue and I can get any setup that I would like. However, there are 2 major limitations to embryonic stem cells. The first limitation, it's immune rejection. Exactly like in donation, we need an immune match. And if I take just embryonic stem cell from any embryo, these cells will be rejected by the body of the patient. And this is a big issue, and we return back to the same problem of organ transplantation. Another major problem is ethical problem. In order to isolate these cells, these embryonic stem cells, we really need to kill an embryo. And of course, some people will say that 64 cells is not a living embryo, but there is a big issues around that. So now I would like to show you how the field and how specifically our lab is dealing with this problem and what kind of technique are present these days in order to generate embryonic stem cell-like cells, but not from a real embryo. The first technique, I'm sure you are all heard about it, is cloning, or in its professional name, somatic cell nuclear transfer. You all know about Dolly the sheep that was cloned. And let me just explain very in a simple way how this technique works. So the scientist who developed this technique called John Gordon, and he showed that if he takes an egg and removes the DNA from the egg, and in the same time take DNA or nucleus from another cells in the body, he can put this DNA into the enucleated egg, give a pulse, electric pulse, and then we start to see exactly the same embryonic development that I showed you before, but this time without any sperm. And what we are actually doing here, we are doing cloning. If I took a skin cell from a sheep and I will put the DNA inside an empty egg, eventually the embryo that I will get will look exactly and behave exactly as for the donor sheep. However, we are not trying to cloning animal here, but rather to generate this blastocyst structure, this early embryo, and then we can isolate embryonic stem cells from the patient-specific cells.
Sucks on the egg. The egg is surrounded by a membrane called the zona, and you'll watch this pipette on the right first drill a hole into the zona, then go in and suck the nucleus out, and then another nucleus, which has been taken from, say, a somatic cell, a cell of the body, is going to be put in. So if we could start the video, please. You'll see now that this drilling pipette is going to drill a little hole into the membrane. You can maybe see a little bit of the hole right here at the next part. This pipette, you'll note, isn't really sharp like a syringe. There you can see a bit of the hole. And now the pipette's going to go in and remove the nucleus. And if you look carefully in the pipette, you'll see a line in the nucleus, which are all the chromosomes lined up. So that nucleus is going to be squirted out now because we don't need it anymore. And there we have an enucleated egg. Now the next step is to take a set of eggs like that, and I'll show you two, and then transfer into them a nucleus from another kind of cell, a fully differentiated somatic cell. So here the enucleated egg is set on the side, that it's held by this holding pipette on the left. There's drilling the little hole in the membrane. Here we go in. Here comes the nucleus from the right. The pipette goes in, and these pipettes are operated with a piezoelectric device, so you can't see.
Okay, so you saw how this technique is working, and since doll is a sheep, as you can see here, many different animals have been cloned, and this technique is really feasible currently even for human beings. So why do we need this cloning technique at all? So beside what I explained in the beginning, this will allow us to generate embryonic stem cells from patient-specific cells. We can also model different devastating diseases. For example, if a person with a Parkinson comes to the clinic, I can take his skin cells, take the nucleus from the skin cells, introduce it into a nucleated egg, generate embryonic stem cells. These embryonic stem cells will force to become neurons, and now I can study about the disease. What caused the disease? What are what can I see that is problematic in these cells? I can run a screen of drugs to see which drug can rescue this problem. So these are really important tools to study different human devastating disease. Another aspect is we can preserve extinct animal species, and recently it was done for the white rhino. People took cells from the rhino body and introduced it into normal rhino, and they were able to clone again this white rhino. Another aspect is for the farm industry. We can enrich for animals that give rise to high quantity of milk or high quality of meat, and so on. And actually, there are other ways to use it. I'm not recommending it, but look on Barbara Streisand.
Are you fighting over the same toy? Really?
Okay, so yeah, if you're really rich and you really love your dog, you can even clone your own dog. But let's return back to science. So now we have this technique that's called cloning or somatic cell nuclear transfer. And this technique solves our immune rejection problem because we take the cells or the DNA from the patient, his own cells. So the body will not reject it. However, we still have two problems. We still have an ethical problem. although this embryo was not generated by sperm and egg, it's still an embryo and we still need to kill it in order to isolate the embryonic stem cell. And we are back to the same problem that it requires egg donation. So this is a second problem of this technique. Now we will go to a second alternative source of cells. How can we generate embryonic stem cell in a different way? And this is based on Shina Yamanaka and Takahashi, two Japanese that showed us in 2006 that one can introduce 4 genes that are active in embryonic stages into skin fibroblasts or skin cells, and after 20 days, these skin cells will become into iPS, which are equivalent to embryonic stem cells. In this movie, you can see these skin cells that are introduced with the 4 genes, and you can see the changes that occur in these cells. It's a little bit hard to see it to a naked eye, but here you can see clearly that the cells start to change and to become embryonic stem cell, exactly like real embryonic stem cells. These 2 discoveries of Yamanaka and John Gordon allowed them to get or to win the Nobel Prize in 2012. And this nuclear reprogramming technique actually resolves all problems. We can take the cells from the patient, so we don't have immune rejection. We don't have ethical problem because we do not generate any embryo, but rather directly converting the skin cells into embryonic stem cell. And we don't need any egg for that. So this is really amazing, and it's really a game changer. However, there is still a big problem in this technique, which is the quality of the cells. In this test that we did in the lab, we showed that while a real embryonic stem cell that I can isolate from the embryo can generate an entire pup. This is a mouse pup, 1 day old. However, different iPS cells that should be exactly like embryonic stem cell generated only retarded embryos or embryos that died during pregnancy. So we have this problem that we need to solve, and this is exactly the main goal of my lab. So we have 2 main aims in the lab. One is to improve the quality of the induced in the iPS cells technique, and at the same time, we develop new paradigm by ourselves to generate other cells that are important for the clinic without going first into the iPS stage. How we are doing that? We are using really cutting-edge technology. We have technology that modifies the genome. We have a way to manipulate embryos, mouse embryos. We are using single-cell technologies, which is the cutting-edge technology the current days. And we have really high-resolution imaging to visualize the changes that we are introducing into the cells. So let me give you some example that we did in the lab, how we really found a way to improve the quality of iPS and to generate other cell types. So in this study, in this work, we really study hard the nuclear reprogramming process, and we identify a network of genes that are activated at late stages of the reprogramming process. Then we said, instead of the genes that the Japanese used, let's use our 4 genes and let's see what will happen. And indeed, when we introduce our genes into the skin cells, we could generate iPS cells, but this time, this iPS gave rise to five-fold more healthy mice compared to the OSKM, compared to the Japanese genes. So this is really a breakthrough in the field that we can identify different genes that can improve the quality of these iPS cells. Another thing that we are doing in the lab, we are converting skin cells into heart cells. If you will look closely here in the screen, you will see that these cells are really beating exactly like heart. So surrounding these beating cells are the skin cells, And these are the cells that we converted into beating cells. So this is a great source for transplantation after a heart attack in order to cover a scar or something like that. This was done by Noah Maoz, a former postdoc in the lab. Another work that was done by Ahmed Radwan, a PhD student in the lab, he used a technique that introduced 3 genes that allow the conversion of fibroblasts into neurons. Here we use a specific color, a green color, that marks for us whether we have succeeded to generate the neurons or not. And as you can see here beautifully, we were able to generate large amount of neurons from these skin cells. But to tell you the truth, my fetish is all about fertility. And we put a lot of effort in the lab to find a way to find solution for fertility problem. Let me give you some example for that. A work that I started during my postdoc, and my PhD student Natalia Stefanenko continue it now in the lab, is to find a way to generate sperm for a sterile man. So imagine a situation that a sterile man comes to the clinic and he really wants kids that will look like him and will behave like him, which means they need to have its own DNA. But this guy doesn't have any sperm. So what we can do is that. So let me tell you what we will be able to do in the future. We can take a skin cells from this guy and then convert them into iPS, as I showed you before. This iPS, we can force to differentiate to become the germ cells. Germ cells are the cells that eventually become sperm. However, these germ cells cannot do it without a support of other cells that called Sertoli cells. So in order to be able to generate sperm in the dish, you need a germ cells, but you need also a Sertoli cells. So this work, as I said, started on my postdoc. We identify a combination of genes that when introduced into skin cells can become Sertoli cells. These Sertoli cells are really functional. What you can see here, the red dots here, or the red circles, are the germ cells. And we culture them together either with control, which is the skin cells, or with testicular extract, or with our induced Sertoli cells. And as you can see here beautifully, our cells could maintain these germ cells much better than any other control. Another aspect is that these Sertoli cells build the tubules inside the testis. So we wanted to see whether they can, when injected into an embryonic testis, whether they can participate in the construction of the tubules. What you can see here are the red cells. These are the native Sertoli cells, and the one that are marked by yellow are the cells that we injected, and you can see beautifully that these cells integrated into these germs, into this testicular tubule. So now we are really close to find a system that will allow the generation of a sperm outside of the body. So if we walked about women problem, what about a women problem? So there are many women that suffer from recurrent miscarriage. Actually, it's 1 couple out of 100 couple. And one of the problem is 70% of the cases is due to placental stem cell abnormality. So Hani Ben Shitrit, a former PhD student in the lab, wanted to succeed to convert skin cells into placental stem cells, and indeed she could do it by identifying a unique set of genes that converted fibroblasts into placental stem cells. These placental stem cells, when injected into donor embryo, and we are talking about mouse right now, about mice, could contribute to the development of the placenta. This is a placenta, and the brown dots that you can see here are the cells that we injected. So this shows that these cells are really functional. Moriana Ama, an MD PhD student in the lab, said, okay, if we can do it with mice, why not in humans? And indeed, recently, we could identify a different combination of factors, of genes, that when introduced into skin cells, can convert them into human placental stem cells. This time, we cannot inject it into real embryo because we are talking about human, and we are still not in the clinical trial aspect. But what Morana Hamim, another PhD student in the lab, and Moriana Ama, they show that this placenta stem cell can generate a mini placenta, which called organoid. These organoids really mimicking the function of the developing placenta, showing that these induced placental stem cells that came from the fibroblasts, from the skin cells, are really functional. So I showed you that we can generate fetal stem cell or embryonic stem cell. We can generate placental stem cell. But what about the third compartment, the yolk sac stem cells? It was shown that if you isolate these three stem cells from an embryo, you really can generate a synthetic embryo. This is how really real embryo look like. And this is how synthetic embryos look like. And you can see that they are really similar. So if we can find a way to generate all the three compartments, all the three stem cells, we will be able to generate a synthetic embryo in the dish. And indeed, Hani and Muhammad Jaber took this job on themselves, and happily, they were able to generate all the three stem cell compartments from skin cells. And here you can see a beautiful illustration how we take these skin cells and push it uphill in order to generate the three compartments.
Hallo. Na, Nathanael, grüß dich.
So this really shows you that it is possible to create a synthetic embryo in the dish. Of course, we are not trying to clone anything or to do crazy stuff. The purpose here is to really study early embryonic abnormality, either in the placenta or in the embryo, to find the solution that will allow us to avoid a retarded embryo or embryo with different problems. Just to show you that these cells are really functional, we took these fetal stem cells, we marked them by red, and then inject them into donor embryo. And if our cells are functional, the embryo should be red. And indeed, as you can see here, the embryo is completely red and the placenta is negative, which means that our cells really contribute to the entire embryo. The same thing you can do with a placental stem cell. We mark them by green, inject them into the donor embryo, and this time you can see that the placenta is green, but the embryo is negative. But this is really functional test. In order to be sure that these cells really convert their identity, we really need to show that their genome, or more accurately, their epigenome, change accordingly. Every cell type expresses a specific set of genes. So we really want to show that this set of genes is specific to the setup that we aimed for. And for that, we use a computational analysis that was performed by Ahmed Radwan, a fantastic PhD student that did this analysis. And what you can see, it's only an illustration, but it shows you the power of this technique. So what we do here, we take the genes that express in any cell type and put them in an abstract space. For example, the skin cells are located here, the real placental cells are located here, and the real fetal cells are located here. And now we are asking, what happened to the genes that are present in our induced placental stem cell and induced fetal cells? And one can see immediately that these cells express the same genes as the real cells. So this is only one very simple example, but just to show you, Ahmed did here a lot of work, a lot of bioinformatic analysis that I will not elaborate in order to prove that these cells are really like the real cells. So in the 5 minutes that I have, I would like to shift gear to the second topic, which is rejuvenation. So how our technique reprogramming is relevant to aging and rejuvenation. So first we need to understand what happened during aging. During aging, we have a specific molecules that decorate our genome. One of them called DNA methylation. And as we age, these molecules decorate the genome differently. So if we can reverse these changes and take this modification back to an early stage, we can actually rejuvenate the cells. And this was shown very nicely by Steve Horvath, who showed that there are 353 sites in the genome that can predict very accurately the age of a person. And if you live an healthy life, your epigenetic clock will show that your biological age is earlier than your chronological age. And this is really amazing. So the first thing I showed you, that if we take skin cells, even elderly skin cells, let's say 80 years old, and reprogram them into either embryonic stem cell or placental stem cells, these cells should be on age 0, because these are embryonic cells. So first thing, we check whether this is true. We took elderly fibroblasts, skin cells, convert them into embryonic stem cell or placental stem cell, and measure the clock that gives us the age of these cells. And indeed, you can see that 32 weeks becomes 0 or 5, and 20 becomes 0 or 3.4, showing you that these cells not only change their identity to a specific stem cell, but rather they were rejuvenated into embryonic stage. However, we don't really want to change the identity of the cells. So if I would like to rejuvenate the skin of someone, or the blood cells, as I showed you, or the eye, in order to restore vision, we want the cell to remain the same cells. We don't want to change their identity. So in order to do that, we decided to stop the reprogramming process after 6 days. This will allow only partial reprogramming. We will maintain the identity of the cells, but we will be able to rejuvenate the cells. And indeed, when we tested it in cells, we could see that even looking on 6 days before the identities change, one can reduce almost by half the age of the relevant cells. But I show you epigenetic stuff, but what about function? Would they also improve did they also improve their function? So here I show you a really important assay. As we age, our DNA damage machinery, the machinery that deal with DNA damages, are really bad. And we start to accumulate many DNA damage regions. And this can be seen here by these purple dots. So when we look on old cells, we can see that on average, we have 6 dots like this in every cell. However, following the rejuvenation process, we can see that before, instead of 6 dots, we can remove it to 1.5 dots in average. This means that these cells not only rejuvenated their age, they also function better. I would like to end this part by showing you that recent work showed that this technology that is complement to our technology could also restore the vision of old mice by rejuvenate the optical nerve in the eye. So I would like to finish my talk by returning back to our vision here. The vision is to generate cells from skin, from patient-specific cells, or really to rejuvenate our immune system in order to prevent diseases that related to aging. Let me just thank the people, and by that I will end up my talk. So first of all, I would like to thank my awesome and really amazing research staff Dr. Kirill Makedonsky, who is responsible on all the embryo manipulation that you saw in this presentation. Shulamit Saban, Dr. Shulamit Saban is my lab manager, and actually she is doing everything. She's orchestrating the entire lab, from guiding students to purchasing all the consumable and to do her own research. And my wonderful group that did all these jobs, all this work. Let me go over the people that I didn't mention in the talk. Azar Yassen is working on modeling placental insufficiency using the reprogramming technique to placental stem cell. Meir is trying to understand how to generate high quality yolk sac stem cells. Idan is trying to generate cells even earlier. He really keen on identifying genes that are important to generate the egg. Rachel is working hard in order to find the solution to generate high quality IPS, and Arij is working to generate these germ cells for the fertility problem. I would like to end my talk by saying thank you very much. And if you find this really exciting and you want to take part in this research, there are three main routes to do that, or you can find your own routes. You can either adopt a student. We really be extremely glad if our student will be, their salary will be covered by donation. You know, technologies is always important, and with time, the technology become more and more expensive. And so if you want, you can donate for equipment or consumable, which is our maintenance, and without we cannot do anything. With that, I would like to thank you all and to end my talk, and I will be happy to take questions. Thank you.
Thank you, Yossi, for this really wonderful talk. That's really exciting. So if anybody has a question, please write it into the chat now. Maybe I'd like to begin with one. You described the way you improved this nuclear programming technique by introducing different types of genes. Are other groups actually using this technique, or has it like gained popularity, or how is it?
So it started by gaining a lot of popularity. However, and we said it also in the paper that we published, that unfortunately this specific combination works only in mice. So from then we are still trying to understand which combination will do the same with human cells. So because it is not really working with human cells, there's no need to take this approach in order to improve the quality of IPS. Once we will identify this specific network in human and will improve human quality, which is much harder to estimate because we cannot generate human beings, then I believe everybody will take this approach.
Okay, thank you. So let me take a question. One person is asking, how does this work actually affect the telomeres? The rejuvenation, I guess that.
That's the question. So the telomere become exactly as young, they become longer and longer, and everything become back as normal. It's become exactly like normal fertilization. So all the aspects of aging restore back to our towards the embryonic stage.
Okay, thank you. Another question that came up is, where do you see the most urgent use of your research? Like, where will it be applied first?
Right, this is a great question because I think about this all the time. And I believe the easiest way to start with clinical trials will be actually, it depends on what we are talking about, whether rejuvenation or, let's start with the rejuvenation. With the rejuvenation, we need an organ that is external to the body, okay? Because we need to introduce the genes, and it will be easiest to do it with an organ that is external, like the eyes. The eyes are external organ. We can just, and this is already was done by many other groups, not our technique, but introducing material into the eye. So we can really restore vision or try to restore vision or to rejuvenate the optical nerve in the eyes. This is something that can start very easily and very fast. You just need to overcome the regulation of safety and stuff like that. Also the blood stem cells. Also rejuvenation of our immune system is relatively safe. We can take just some blood from patient, manipulate the stem cell inside and transplant back this blood, rejuvenated blood, into the same person. If you are talking about the regenerative medicine, once again, it will be easiest to implement when we don't need a 3-dimension organ, okay? Because 3-dimension require many different cell types. So again, we are returned back to the blood system, okay? Anyone that need blood stem cell because he had cancer and he did a very strong chemotherapy that erased or destroyed his bone marrow, so we can generate blood stem cell for him to repopulate his immune system, or even Parkinson's disease that require just the dopamine to be secreted from the right place. So if we will be able to generate dopaminergic neurons that are fully functional and inject them into the right area, then I believe it will at least partially rescue or delay the disease, the progression of the disease.
Okay, thanks. Just maybe a follow-up question. You showed the placental stem cells, and which is actually pretty far along in their development. Where do you see, like, when will the first clinical trials be ready? What's your guess?
So this is really, it's like a future medicine. And first, the people need to understand, they need to cross the bridge in their mind that this is possible, and this is how the medicine will look like in 20, 50 years from now. So once I will pass this bridge, I believe everything will move forward much faster. So currently, our only limitation is to find the company in the industry that will be able to take the risk and invest the money for clinical trials and to do some more research stuff in order to generate the best paradigm, the best protocol to move forward to the clinic.
Okay, maybe just a quick, clear question. What does it mean to change the identity of a cell? Maybe if you can just briefly explain it.
Yes, so we have more than 300 cell types in our body. We have blood cells, we have kidney cells, we have liver cells. Each cell has its own identity, and how the identity is determined by the genes that are active in every cell type. We have around 20,000 to 25,000 genes in every cell, but every cell type activate only a fraction of these 25,000 genes. Heart cells will activate one set of 10,000. Liver will activate different set of 10,000 genes. So what we are doing in reprogramming, we erase all the genes that are active in skin cells, and we deposit new genes that are relevant to the cell type that we want to convert to. For example, if it's embryonic stem cell, it allows activation of genes that are important for the embryonic stem cell. If we want placental stem cell, this reprogramming technique activates the 10,000 genes that are important for the function of the placental stem cells.
Okay, thank you. So there's one, somebody has a very specific question. The technology has changed dramatically over the last few years. What future improvements in DNA technology do you see as necessary to improve your research forward?
Right, so we, as many labs around the world, are using genomic manipulation technique. And now I'm sure most of you heard about it, the CRISPR-Cas9 technique that won the Nobel just recently. And we are using this technique. And currently, we know how to change mutation, how to introduce missing fraction into the genome. So if we know on a specific mutation or a fraction in the genome that is problematic and need to be corrected, we know how to do that. Of course, there are still some limitations, like off-target effect, but this can be overcome relatively easily using either bioinformatic technique, bioinformatic analysis, or eventually even to sequence the entire cell to see that all that the entire DNA is intact.
Okay. Then we have more of an ethical question. As always, there are positive intentions in research, but what if somebody has negative intentions? What if a criminal with financial resources needs a liver transplant? Will he be able to find a lab to do, let's say, things that aren't ethically okay?
I'm afraid that I believe he will find a lab somewhere in the world. I will not mention the country. But for that, we have tons of ethical committees that deal with it all the time. But I'm sure if you heard, in China, for example, there was a scientist who modified the genome of a baby, actually 2 babies, of a mother that was sick with HIV. He wanted to show that when he modified a specific gene, these babies will not be sick with HIV, although the mother is sick. And eventually, this is a very sad story because these scientists went to jail eventually, and these babies, although they were they were not sick by HIV, some scientists believe that they will live, their lifespan will be shorter. They might be more smarter, maybe it's a good thing. But currently, I'm afraid of genomic manipulation. We don't know everything, and manipulating genes just like that is really bad. But this is in contrast to correcting a mutation. When we are correcting mutation, we know exactly what is a problem. We know what we need to fix. And here I don't see any problem in doing that.
And how is it in your work at the Hebrew University? Are you actually dealing with ethical issues? Do you spend a lot of time, I don't know, getting ethical approval for, I don't know, the development of the synthetic embryos?
So we do have ethical committee all the time, and we need to apply many reports about what we are doing, and there is ethical committee that discuss it and decide what are the best control and what we need to do in order that it will be approved. But currently, we are fine with that because most of our proof of principle is done with mice. So for that, it is a little bit easier to get an ethical approval, but still, we need to write a lot of ethical report to convince the committee that what we are doing is really important and it will lead eventually to saving life and not just to playing with mice.
Okay. One more question that I'm not really familiar with, but can the technology also apply to cryogenetics to repair frozen human tissue?
It all depends. Yeah. So if we are talking about tissue, there is always an issue about delivery. When we are talking about cells in culture, the delivery is relatively easy, But once we are moving into a living tissue, the delivery aspect become really an issue here. And for that, we need to improve the field, the entire field need to improve its delivery capabilities. There are some diseases that you don't need to correct all the cells in order to slow down a specific disease or even rescue a disease completely. In these cases, even 10% correction, which is possible in several organs, will be enough to slow down a disease or even to rescue a specific disease.
Okay, thank you very much. I think there are no more questions and we are spot on the time. 7 o'clock. Thank you very much, Yossi.
Thank you all. Thank you, Nadia, for the invitation.
Thank you.
And I hope you enjoy the lecture.
It was very interesting.
Thank you very much for this comprehensive talk. And now we know a lot more about your techniques and how one could combat disease and problems.
Thank you very much, Nadia.
Yes, thank you very much. It was a great lecture.
Fantastic.
Thank you, Yossi. And I see that a lot of your students are here with us.
Yes.
So
maybe you should ask the student also question. Let's see how ready they are.
So if you want to open your microphone to the students, that I see all of them together there. So thank you to you too.
Hello.
Great.
They are very supportive. You see, they stayed late to hear their the head of the lab giving a talk.
So it seems like a great team.
Yeah.
Yeah.
Definitely.
So I might get back to you as a science journalist because it was very interesting.
Sure. Okay, so are we done, Mara?
I think so. We are done. And as I said, we'll receive the recording in a few days. And if you have any more question, I think you can write Nadia or me or Yossi.
Sure.
We are open to answer to all your questions.
Thank you all and bye-bye.
Thank you very much, Yossi.
Thank you.
Have a nice evening.
Bye.
Yes,
Thanks.
Bye-bye.
Bye.
Bye-bye.